So welcome back to Gynae Wizard, and today we'll be discussing about the fourth episode in the placental series, that is the placental function. So in the placental function, there are many functions of the placenta. So today we are interested about the nutrient transport and the gaseous exchange, which we have already discussed in the couple of question and answer series on my channel. So what do we mean by nutrient transport? So we all know that the fetus gets all its nutrients from the maternal circulation. So placenta acts like a mediator between the mother and the fetus. So there is transport of glucose, fatty acids, amino acids, and many other micronutrients between the mother and the baby. So this is mainly essential for the fetal growth and development throughout the pregnancy. So this we all know. So what do we mean by nutrient transport, and what are the main important points which we have to remember with respect to the gaseous exchange? We will discuss in today's class. So the firstly, I already mentioned that placenta has two types of functions. One is the anabolic and the second one is catabolic. So the first half of the pregnancy is usually the anabolic type of function which the placenta performs. So it stores all the nutrients, and mainly in the first half of pregnancy, what the main function of placenta is mainly for implantation. So implantation occurs, so the trophectoderm goes and converts into the placenta, the cytotrophoblast, syncytotrophoblast, everything is formed and the implantation occurs. So all this will be occurring in the initial first trimester, and also many other hormones are released from the placenta, which is very important, and placenta also acts like an important endocrine organ during the pregnancy. So first half of the pregnancy is considered to be anabolic, where other functions like implantation, endocrine function play an important role. So in the first half, there is building up of all the energy sources, and there is buildup of the glucose in the form of glycogen, and even the fat deposition occurs in the placenta. But the second half of the pregnancy is mainly catabolic in nature. So why is it catabolic? Because as the pregnancy advances, the fetal growth will go on multiplying. So the hyperplasia and the hypertrophy of the cells in the fetus is more, and even the weight which the fetus gains during second trimester and third trimester increases. So this is mainly because of the catabolic nutrients which are supplied from the mother to the baby. So what happens? There is increase in insulin. So we have all studied, when we are studying gestational diabetes mellitus, we have gone through that during the 24 to 28 weeks of gestation, there is increase in insulin and also called as hyperinsulinemia. So why this hyperinsulinemia occurs? So this hyperinsulinemia will promote the continuous shift of glucose from the mother to the baby, so that the baby is not deprived of any glucose, and the baby carriers of all functions with the help of the transfer of glucose from the maternal circulation. So that is why the energy is mobilized from the maternal fat stores even in the fasting state. So even in the fasting state, when the glucose is not readily available, that time the maternal fat stores get broken down, and these fatty acids are supplied to the baby. So that is how the baby is not deprived of any nutrients irrespective of the mother's condition. So whatever might be the mother's condition, the baby has to have continuous supply of the nutrients, and this is mainly facilitated by the placenta. So today we will discuss each and every nutrient and how is it catabolized and supplied to the baby. So as I told you in the earlier slide, so this is a pictorial representation of the function of the placenta during pregnancy. So until the week 12, we have implantation, we have perfusion with the maternal blood, and we have the first fetal blood cells and the endothelial cells which start the formation of fetal vessels. And also there is immunotolerance, so that is why the fetus is not rejected, and immunological adaptation also occurs in the first 12 weeks. And this transport of glucose, so that is why the first 12 weeks is regarded as the anabolism, where there is development of the placenta and everything falls in place. But what happens in the week from week 13 to 26 and week 26 to 27 to 40, that is in the second major half of the second half of the pregnancy, there is catabolism. Why there is catabolism? Because this will ensure the rapid growth of the fetus. So what is necessary for the rapid growth of the fetus? The transport of glucose, amino acids, fatty acids, and also some of the trace elements like zinc, magnesium are also necessary for the fetal growth and brain development. And gaseous exchange should definitely be important because the fetus directly depends upon the placenta for oxygenation because the lungs are non-functional with respect to gaseous exchange. The lungs are developing, but the alveoli are collapsed, and that is why the gaseous exchange will not happen at the level of the alveoli, and the fetal circulation plays a main role and helps the gaseous exchange to take place. So this is why the second half of the pregnancy is regarded as a catabolic state. So firstly, among the nutrient transport, we are interested in glucose, fatty acids and amino acids. So firstly, we will discuss about the transport of lipids in the form of fatty acids. So these lipids are very much important, and especially even in the maternal fasting state, these maternal lipids are mobilized in the form of fatty acids and they supply the fetus. So how does this occur? So firstly, we have to remember that in the first half of the pregnancy, because it is anabolic in nature, there is maternal hyperlipidemic state. So maternal hyperlipidemia occurs in the first half of the pregnancy, and mainly this is because the mother has hyperphagia during the first 3 months. Though there is nausea and vomiting, but the cravings and the satiety is not achieved, so that the mother feels like eating more. So this hyperphagia is a reflex for the development of hyperlipidemic state of pregnancy, especially in the first half, because it is an anabolic in nature. So what happens? There is buildup of the maternal fat stores which occurs in the first half of the pregnancy. So these fats are available for the fetus in the second half of the pregnancy. So this is very important for the fetus for its growth and even the formation of fetal fat stores. So the fetus also has to have its own stores for its usage. So this is very important. So in the first half of the pregnancy, the mother has hyperphagia, which results in the hyperlipidemia of pregnancy, and there is deposition of fat in the mother. So that is why there is maternal fat stores which are building up. But in the second half of the pregnancy, these maternal fat stores are broken down and these are facilitatedly transported to the fetus, and this establishes the formation of fetal fat stores. So how does this occur is the question. So we all know that the fatty acids are transported from the maternal circulation to the fetal circulation, but how is the main question mark. So firstly, we have to understand that the free fatty acids are only available for transport, because only free fatty acids can transfer or can cross the placenta, because if the fats are esterified in the form of triglycerides or phospholipids, they cannot cross the placenta. And amongst the free fatty acids also, only 1% are free, but all the 99% of the free fatty acids are bound to lipoprotein. So these has to be broken down and the essential free fatty acids have to cross the placenta. So these free fatty acids are the building blocks for the establishment of both maternal and fetal fat stores. So that we have to remember. So amongst this, so 99% are bound with lipoproteins. So that is why at the level of the placenta, we have to break down these lipoproteins and we have to carry the free fatty acids and supply to the fetus. So essentially there are two processes, because the free fatty acids only can cross the placenta. The provision of fatty acids, the provision of fatty acids at the level of placenta occurs through two processes. So this is the presence of two types of lipases which are present. So this lipases, one is intracellular lipases and the second one is extracellular lipases. So the intracellular lipases are present in the syncytiotropoblast. So these intracellular lipases, which are present inside the layer of syncytiotropoblast, they take the triglyceride-rich lipoprotein particles. They cause hydrolysis of the free fatty acids, and then they transfer it to the fetus. So how does this occur? The syncytiotropoblast has something called as lipoprotein receptors, which are present on the syncytiotropoblast. So as I told you in my first video, syncytiotropoblast is like a hero. So it does so many functions. It has endocrine function, it has immunological function, it has this nutrient function, but the syncytiotropoblast is highlighted. But the thing which supplies the syncytiotropoblast is the cytotrophoblast. So equal role of both is important. So the syncytiotropoblast has these lipoprotein receptors, and these triglyceride bound lipoprotein particles are bound to the lipoprotein receptors over the syncytiotropoblast, and then they are acted over by the intracellular lipases, and these fatty acids are released and they are released into the fetal circulation. So this is one process. The second process is the extracellular lipases, which are present in the villus space. So these lipases, they go and act on the these triglycerides and the phospholipids, and they release the free fatty acids, and these free fatty acids are directly transported into the syncytiotropoblast. So extracellular lipases, they go and act, and the free fatty acids are taken inside the syncytiotropoblast, and some of the molecules are bound to the receptors which are present on the syncytiotropoblast. They enter inside the syncytiotropoblast, and the intracellular lipases will go act on them, and finally the end product is release of free fatty acids. So these free fatty acids immediately in the placenta again are esterified into phospholipids and triglycerides. So this forms the placental stores. So once they form the placental stores, they are preferentially shunted into the fetal circulation. So these free fatty acids are preferentially shunted. So what do you mean by preferential shunting? So whenever there are many free fatty acids which are entering from the placental reserve to the fetus, but what is favored is the essential free fatty acids. So what are the essential free fatty acids? Omega-3 fatty acids, omega-6 free fatty acids, and polyunsaturated fatty acids, especially the docosahexaenoic acid. So all these essential free fatty acids are preferentially shunted from the placenta to the fetus. So that is why whenever you compare the concentration of docosahexaenoic acid in the maternal plasma and the fetal plasma, the fetus has more of DHA when compared to the maternal circulation because of this preferential shunting. And this is very much required for the optic nerve development and also the brain development of the fetus. So that is why this free fatty acids are very much essential for the fetal growth and development, especially the neural development mainly occurs with the help of free fatty acids. So secondly, we have to speak about the transport of glucose. So transport of glucose is mainly by facilitated diffusion. So what do you mean by facilitated diffusion? So the concentration in the fetus is less. The concentration in the mother is more. So that is why there is transfer of glucose preferentially from the mother to the fetus. And we all know that the glucose is an essential source for the basic requirement or the basic need of the fetus for energy production. It requires glucose. That is why there must be continuous supply of glucose from the mother to the baby, and that is why it is called as facilitated diffusion. And why is it called as facilitated, and what is facilitating the transfer of glucose is mainly the transport proteins. So what are the transport proteins? We all know that it is the GLUT transporters. So GLUT transporters are very much essential, and these facilitate the transfer from the mother to the placenta and from there to the fetus. So what are the transport proteins which are present? It is the GLUT1, GLUT3 and GLUT4. So GLUT1 is mainly present on all the different cell types on the placenta and fetal cells. And GLUT3 is mainly present on the fetal endothelial, and also it is present on the stromal cells of the placenta. And lastly, GLUT4 is present only on the exclusively on the placental stromal cells. So these transport proteins are very much important for the transfer of glucose from the mother to the baby. And this GLUT transporters, they have a high capacity, but this transfer is not regulated by anything. It is an unregulated and unlimited process. That is why whenever there is gestational diabetes mellitus, whenever there is maternal hyperglycemia, there is increase in transfer of the glucose from the mother to the baby. And this results in fetal hyperglycemia. And when there is fetal hyperglycemia, there is stimulation of insulin production, and this results in fetal hyperinsulinemia. And that is why there is macrosomia, and there is increase in the enlargement of the long bones, and this also might be one of the reason for shoulder dystocia during birth. So that is why this is unlimited. So if the mother stores are very much, this will be transferred to the baby. If it is less, it will not be transferred. So that is why this is very much unregulated and unlimited, and that is why this is a little problematic to the fetus. Because if there is a regulation, so irrespective of the amount of glucose which is present in the mother, only this much of glucose should be transferred. If there is some mechanism like that, it should be regulated. But unluckily, we don't have such regulation in the placenta or in the mother. So that is why how much ever there is glucose, surplus amount of glucose in the mother, it is continuously being transferred to the baby. And this is regulated, and this has to be available for the fetus for the fetal growth and development. So essentially by term, at least 5 mg of glucose per kg body weight per minute is transferred to the baby. It is transferred to the fetus. But one important thing you have to analyze, that all the maternal glucose will not reach the fetus. So half of the maternal glucose will be present in the placenta, and the placenta also is a very metabolically active organ. So it will convert the glucose into lactate, and it will form some of the uh, placental stores in the form of glycogen or lactate. Only other half of the glucose is supplied directly to the fetus. And there is something called as the oxidative glucose quotient. So this glucose quotient in humans is only 0.8. So that means that the amount of glucose which is available for oxidative metabolism is only 0.8. So that is why the fetus depends on other sources also for oxidative metabolism, like fatty acids or amino acids or lactate. So it depends upon other sources also for oxidative metabolism, and glucose is not the only nutrient which is essential for oxidative metabolism. So that is why glucose is also necessary, but other protein transfer is also necessary. And that is when we have to discuss about the carbohydrate metabolism during pregnancy. So the carbohydrate metabolism will have 3 pillars. So one is the fasting hypoglycemia, postprandial hyperglycemia, and insulin resistance. So why these are important? These are important for the continuous transfer of glucose from the mother to the fetus, because fetus is entirely dependent on the mother for its glucose stores, because fetus uh, cannot uh, fetus is dependent and it cannot produce glucose as such. It is dependent on the mother. So these three pillars will help in continuous transfer of glucose from the mother to the fetus. So whenever there is fasting, whenever the mother is fasting, what happens? The fat stores are mobilized, and we already discussed that in the form of free fatty acids, they are supplied to the fetus. But once the mother takes food, there is postprandial hyperglycemia. So why there is postprandial hypoglycemia? Because there is underlying insulin resistance. Why there is insulin resistance? That we have already discussed, because there are many anti-insulin factors which are present and which are secreted during the pregnancy, especially the HPL, human placental lactogen, which is secreted by the placenta, and this also has an anti-insulin property. And insulin is enzyme also, which is secreted from the placenta, also has insulin resistance property. And whenever there is insulin resistance, there is postprandial hyperglycemia. So what do you mean by insulin resistance? Whenever there is insulin which is secreted, it will allow the glucose to go and enter the metabolically active organs like muscle, liver, and it will convert it into the stores like glycogen or lactate. But if there is aerobic metabolism, it will be converted into glycogen. If there is anaerobic respiration or metabolism, it will be converted into lactate. But this is not facilitated. So the conversion of the glucose into the stores is not facilitated because there is underlying resistance. So that is why there is shift, or there is shunting of all this glucose from the mother to the fetus. So that is why these three are very much important for the transfer of glucose from the mother to the baby. So these are the main pillars of carbohydrate metabolism, and that is why the mother is under the risk of development of gestational diabetes mellitus during pregnancy. And that is why we have to screen her once again during 24 to 28 weeks. Once we have screened her in booking, but again we have to screen her between 24 to 28 weeks of gestation, because there is surplus amount of anti-insulin factors which increases the insulin resistance and which might unmask the development and unmask the glucose intolerance during pregnancy and results in GDM. So that is why we have to screen once again, and this is the importance of carbohydrate metabolism. So next, coming to the protein transport. So what are the building blocks of proteins? We all know that the amino acids are the building blocks of protein. So these amino acids have to be transported from the mother to the baby, and this is mainly facilitated by the transporter proteins which are present in the syncytial trophoblast. And we have to understand that there are two types of transporters. One are the accumulative transporters and one are the exchange transporters. So what do you mean by accumulative transporters? These accumulate the essential amino acids. They accumulate the amino acids in the placenta and they are shunted into the fetal circulation. But what's the exchange amino acid transporters? These exchange the essential amino acids, and these essential amino acids are preferentially shunted into the fetal circulation. So as fats also. So we thought essential fatty acids like docosahexaenoic acid are essentially shunted to the fetus. The same way, this exchange transporters of amino acids, they essentially shunt the essential amino acids into the fetal circulation, and these are also very important for the fetal growth, especially the neurological development. So how does this occur? So it occurs in three steps. So first step, three steps. So the first step is at the level of the trophoblast. So the uptake of amino acid at the level of the trophoblast. So this is occurring at the level of microvillus membrane. So whenever this is the villus, so these amino acids are entering from the microvillus membrane, and then they are going into the cytoplasm of the syncytial trophoblast. So the cytoplasm of the syncytial trophoblast are uptaking the amino acids, and from there, here there are the fetal vessels. So this are going inside the fetal vessels through the basement membrane. So that is how the amino acids are transported from the intervillous space into the trophoblastic tissue, into the cytoplasm, and then the fetal circulation. And these transporters, both the accumulative and the exchange transport, facilitate the essential amino acid transfer from the mother to the fetus. And next, there are certain trace elements and minerals which are important for the baby as well. So firstly, iron. So iron is very important for the fetal blood development. So that is why iron is always derived from the mother. So the hemoglobin of the fetus wants iron for the oxygen binding. So this iron molecule is also available by the mother. So we all know iron is available in the ferric form and it is bound to transferrin. So transferrin is the iron protein which is bound. It's a transport protein. So whenever this iron, ferric form of iron with the transferrin molecule, so these are the bounded molecules and these are going and they go and attach to the DMT, divalent metal transporter protein, which is present on the syncytium. So this is drawn into this complex is drawn into the syncytium by this receptor, that is divalent metal transporter. And here, before going here, this ferric form has to be reduced to ferrous form. So only then the ferrous form will get entered into the divalent metal transporter. So this occurs with the help of some proteins called as steep family proteins, which cause reduction of ferric form into ferrous form. And this divalent metal transporter proteins are present on the syncytium and they are transported intracellularly, and then they go into the placental stroma, and from the placental stroma they enter the fetus through something called as ferroportin. So this ferroportin transporter transports the iron from the ferrous form into the fetus through ferroprotein receptor. So this again immediately once it enters the fetus, it gets again oxidated to ferric form and then again bound to transferrin and it goes the target site. So this is about iron metabolism. So other trace elements are calcium. So calcium is also very much important and there is something called as the sodium calcium pumps which help in the shift of calcium from the mother to the baby. And there are other forms like there are other elements such as magnesium. So there is something called as a magnesium transport protein and there is phosphorus and there is sodium mediated phosphorus transport and even there is zinc. So all these elements are very much important for the baby. So the fetal growth, development and all some of the minor metabolisms require these trace elements for their function, and as the enzymatic processes occur with the help of these trace elements, even these are transported from the mother to the baby through various transporters and pumps. So this is the slide which we already discussed. So this is like it is a schematic diagram of the same. So glucose is mainly transported with the help of GLUT1, GLUT3 and GLUT4. So GLUT1 is present on all cell types. GLUT3 is preferentially present on the placental stromal cells and the fetal endothelial cells, and GLUT4 is exclusively present on the placental stromal cells. And then we have amino acids, where amino acid transporters, especially the accumulative and exchange transporters, play a role. And next, coming to all the lipid molecules. So lipid molecules have to be broken and they have to be non-esterified to fatty acids, and then only they can be taken up by the placenta, and placental fat stores are formed and again they are transferred to the fetus, and preferentially the essential fatty acids and essential amino acids are transferred to the fetus. So lastly, in today's discussion, we will discuss about the gaseous exchange, which also is one of the very important function of the placenta. So this already we have discussed in our group. So we will just revise it and recall it. So we have understood something called as a double Bohr effect. So what do we mean by double Bohr effect? So before knowing about double Bohr effect, we have to know what is Bohr effect. So what is Bohr effect? So whenever there is increase in the carbon dioxide concentration or decrease in the pH, what will happen? There is decrease in the affinity of hemoglobin to oxygen. So there is unloading of oxygen from the hemoglobin, and that is why the oxygen transfer will occur. So simply to tell that there is increase in carbon dioxide or decrease in pH, that is the hydrogen ions will increase. That time the hemoglobin binds less tightly to the oxygen and more of oxygen is released into the tissues. So this occurs both on both sides. So the Bohr effect occurs both on the maternal side and the fetal side. So firstly on the maternal side, so what happens on the maternal side? The carbon dioxide from the umbilical artery will diffuse into the maternal blood. So whenever there is diffusion of the carbon dioxide from the umbilical artery to the maternal circulation through the placenta, what will happen? There is increase in carbon dioxide and decrease in pH. So whenever there is decrease in pH of the maternal blood, what happens to the oxygen dissociation curve? It shifts to right. So whenever it shifts to right, the maternal HB releases more of oxygen. So this oxygen is very much important. So this is how oxygenation occurs. And what happens on the fetal side? So what happens on the fetal side? There is loss of carbon dioxide because more of carbon dioxide is diffusing into the maternal side. So what happens? There is decrease in oxygen and there is increase in pH. So that time the fetal oxygen dissociation curve moves to left. So what happens? It increases the affinity of hemoglobin to oxygen. So that is why the fetal hemoglobin binds to oxygen more tightly. So this is the main principle or the mechanism how the oxygenation occurs on both sides. So that is why it is called as a double Bohr effect. The second important point with respect to gaseous exchange is that the carbon dioxide is more diffusible when compared to oxygen. So at the placental interface, what happens? The carbon dioxide diffuses more readily from the fetal blood into the maternal blood. So this will promote the oxygenation to takes place more readily at the level of placental interface. So firstly, we have the double Bohr effect. The second thing, the carbon dioxide is faster in with respect to diffusion. It is more 20 times more diffusible, and that is why the unloading of carbon dioxide happens readily and loading of oxygen happens readily at the placental interface. And the third important thing is the Haldane's effect. So what is Haldane's effect? So this all we have read in our physiology and we have forgotten. So how do we apply that with respect to placenta? Is that the oxygenated hemoglobin has less affinity, or it has less affinity towards the carbon dioxide, or it is not able to carry the carbon dioxide. So whenever the maternal hemoglobin releases more of oxygen, what will happen? It will become deoxygenated. So whenever the deoxygenated HB is present, it has more affinity to carbon dioxide. So the deoxygenated HB will carry more carbon dioxide, and this will facilitate the uptake of fetal carbon dioxide by the maternal hemoglobin. So I will repeat. So whenever there is oxygenated hemoglobin, this will have less affinity to carbon dioxide. The carbon dioxide. So whenever the maternal hemoglobin readily it releases the oxygen to the fetal circulation. So it becomes deoxygenated. So whenever it becomes deoxygenated, it has higher affinity for carbon dioxide, which is readily unloading from the fetal side. So that is why it is carrying more of carbon dioxide from the fetal end. So what happens? There is unloading of carbon dioxide and loading of oxygen, which happens at the fetal site. So this helps in oxygenation, and this oxygenated blood is readily carried through the umbilical veins. So this is also very much important, and this Haldane's effect is very much important. And lastly, as we mentioned, HbF is very important for facilitation of oxygenation. So it is not only the Bohr effect or the double Bohr effect or Haldane's effect. The fetal hemoglobin, it has reduced binding to 2-3 BPG. So that we know. But why this, whenever it has reduced binding to 2,3-BPG, it has higher affinity to oxygen. So whenever there is increased affinity to oxygen, it carries the oxygen more readily, and the oxygen dissociation curves shift to left. So even at the lower partial pressure of oxygen, there is increased affinity of oxygen to the fetal hemoglobin, and that is very much important for the fetal oxygenation to takes place. So these are the four important reasons why maternal transfer of oxygen to the fetus occurs readily. So one is the Bohr effect, the second one is the Halden's effect, the third one is the carbon dioxide is 20 times more diffusible, and that helps in the unloading of carbon dioxide, and the fourth one is the hemoglobin, fetal hemoglobin has increased affinity to. So instead of fetal hemoglobin, if there was some HbA, that time the oxygen affinity would have been decreased, and the oxygen loading also would have been hampered at the level of the partial pressure of the oxygen, which is present at the placental gradient, and that would result in fetal hypoxia, which would be chronic, which might lead to fetal growth restriction. So that is why we are all blessed with evolution, that fetal hemoglobin is present, and this has increased affinity to oxygen, and which will facilitate the fetal oxygen to take place readily. So with this will end today's session. So we have discussed the various roles of placenta with respect to the nutrient transfer and gaseous exchange, and this completes my placental series episode number 4 with respect to placental function. So thank you. Happy learning. Keep watching my WhatsApp channel. Thank you.